All right. So I had class today with the B group, and I noticed we're having some issues with some of our concepts. So just a real quick review video. We've talked a lot about different types of transport in the cell. And remember, all types of transport help us to maintain homeostasis. Remember, homeostasis is maintaining a stable internal environment in the cell. We want our cells to be stable. We can't have them going up and going down in the amount of water or the amount of nutrients or getting bigger or getting smaller. We want to stay at homeostasis at all times. So how do we do that? Well, we have two ways. One way is called passive transport. There's three types of passive transport: diffusion, facilitated diffusion, and osmosis. Okay? Now, how are these three things different? So let's talk diffusion first. And we'll just call it simple diffusion. I have a cell membrane, and remember the cell membrane is made of my phospholipid bilayer, right? So it's a bunch of these things. They have a hydrophilic head and a hydrophobic tail, and it surrounds the whole cell, and it's selectively permeable. All this is super important when we're talking about cell transport or just cells in general, okay? So this phospholipid bilayer is selectively permeable. So with passive transport, we go down the concentration gradient. We're moving down from an area of high concentration to an area of low concentration. So the two things that we talked most about that can cross the membrane through simple diffusion were oxygen and carbon dioxide. Okay? So if I have oxygen, let's say the red is oxygen. Okay? There's a bunch of oxygen outside the cell, and I know I'm going up into my writing there. Bunch of oxygen outside the cell, very little inside the cell. What are these things going to do? They can go straight through these phospholipid layers into the cell until we reach homeostasis, okay? Or we get to equilibrium. Equilibrium would be the same on both sides of the cell. So oxygen, O2, and carbon dioxide are both going across the cell membrane through simple diffusion, okay? Without needing a protein, without needing any energy from the cell. Okay, that one's pretty simple. That was like our food coloring. When I put the food coloring in the water, it spread itself out. The other type of diffusion is called facilitated diffusion. Now, facilitated diffusion, and I'm not going to draw all the phospholipids this time, but let's just say this is my cell membrane. And remember, I'll draw one here. Here's a phospholipid bilayer right there, okay? Facilitated diffusion is going to use this thing called a transport protein, okay? And these things are going to have little channels through them that allow molecules to travel through the cell membrane. Now, what kind of things get through the cell membrane through facilitated diffusion? Things like sugars or ions. Ions would be things that have a positive or a negative charge, all right? So if I have a bunch of sugar out here, outside the cell, it's still going down the concentration gradient, okay? And remember, we watched a video on this. It's on your PowerPoint. These things will travel into this little channel and go through the protein channel to get into the cell. Or it may be getting things out of the cell. And they're going to do that, again, until we reach equilibrium, EQU, equilibrium. Same on both sides of the membrane, okay? So facilitated diffusion, the only way it's different than simple diffusion is that molecules are going through a protein channel to get into and out of the cell. Our third type of passive transport was osmosis. Now this was our egg, okay? So osmosis. Osmosis is the diffusion of water. So we had three scenarios. If I take an egg and I put it into, remember, all living things have cytoplasm that are solutions, okay? So I'll just put two scenarios on this board right now. There's one scenario, here's another scenario. Okay, I'm gonna put this egg into a hypertonic solution. This is like when we put the egg into the corn syrup, okay? Now, what we're saying with this, with osmosis, is that these molecules are too big to get through the cell membrane. So how could we reach equilibrium? Well, we would want water to travel out of the cell to reach equilibrium, okay? What would this eventually turn into? Well, we'd have this shriveled up cell that still has those solutes in it, but it's gotten rid of a lot of water trying to get to equilibrium. If you remember, our corn syrup actually turned kind of runny when we finally looked at it, because a lot of water had gone from the egg into the corn syrup. Now, what happens if I take an egg and put it into so this was a hypertonic solution, high amount of solutes. What if I put it into a hypotonic solution? Okay, low amounts of solutes. Well, let's put a couple solutes out here. Well, now there's more water outside the cell than there is inside the cell. So what are we going to do? Water is going to travel in. Okay, what's that going to lead to? It's going to lead to a big swollen cell that's gained a lot of mass because the water has flowed into the cell trying to create again, equilibrium, or make it the same on both sides of the cell. All right? We talked about a couple real-life examples of this. If I take a plant cell and put it and surround it with salt water, it'll shrivel up. That was called plasmolysis. Plasmolysis. The cell membrane has shrunk. Let's pretend this is a plant cell. I'll draw the cell membrane around it, kind of like it's in a box. Excuse me, the cell wall. The cell membrane has shrunk away, and that's when the plant will wilt. We also said, what if you drink salt water? Okay, this situation would occur. In this one down here, this was like the radio contest we talked about. But if the cell fills up with water and maybe it bursts, that would be cytolysis, okay? Cytolysis is when water rushes into the cell and the cell would actually burst or, explode because too much water has rushed in, okay? So again, osmosis caused our eggs to lose mass. I put the one egg into the corn syrup, which was hypertonic, that caused it to shrink. Plasmolysis is a plant cell term, so it's specific to plants, okay? Cytolysis, C-Y-T, cytolysis is when a cell explodes or ruptures because too much water has rushed in because you've placed it into a hypotonic environment. Our last example of passive transport, well, it's still osmosis, but what if I take a cell and I put it into an isotonic solution? Okay, so it looks like the solutes are the same concentration on the inside and the outside of the cell membrane. Well, this thing's going to stay the same side, and water is going to be going in and out of the cell at the same time, okay? We want our blood cells to be surrounded by an isotonic solution, okay? And what controls that for us? What takes care of that for us? Our kidneys. Remember, we talked about how our kidneys go through osmosis and maintain a water balance in our cells. Another example of that is fish. Fish have gills. Gills keep their cells in isotonic solutions, okay? So that's a review of passive transport.